welcome to our presentation on primary ciliary dyskenesia. Today I will be presenting Nicole Hindley and
Catherine Ogden,
George Adams,
and Ashia Udin.
Hope you enjoy it, and we'll leave it over to George to introduce the topic.
Primary ciliary dyskinesia, or PCD as it will be referred to throughout this presentation, just to make things a little bit easier, is a rare disease which will affect 1 in 16,000 people. Now, the disease itself is an autosomal recessive disease. This means that in order for someone to have this disease, they must inherit two of the faulty alleles of the same gene from both their mother and their father. Now, the disease mainly affects the cilia which line the respiratory tract, as well as the fallopian tube, and it also affects the flagella of certain cells like sperm cells. Now, because of this, it leads to symptoms like coughing and gagging, but also severe symptoms like respiratory tract infections, as well as infertility, which mainly occurs in men who have PCD. Now, whenever there is a change at the DNA level which can cause a problem with a protein that is produced, and this problem can lead to the protein being abnormal and therefore not being able to complete its function. With this occurring in the cell, therefore the cell itself cannot perform the correct function, therefore leading to the symptoms we see. The main mutation we will focus on today is the DNA H5 mutation.
So following on from what George was saying about the cilia, I'm just going to describe a bit of an outline of the cilia and what their function is. As illustrated here, these little orange protrusions are cilia, and they are slender hair-like structures that are on the surface of certain cells in the body. For example, cells lining the trachea, which is your windpipe, have cilia on the surface, like this diagram here, in order to brush mucus up and over the top. Mucus is released by a goblet cell and then secreted onto the surface of the trachea cells, and then the cilia's job is to bend like a brush and sweep the mucus up and out of the trachea. Also, similar process in the fallopian tubes, which is the tube from the ovary to the uterus. The cilia in the fallopian tubes, their job is to brush the egg down the fallopian tube into the uterus, ready for fertilization. And George mentioned flagella as well, which is a similar structure to cilia. And flagella is present on sperm cells, and its job as well is to move, so it creates more of like a whip-like motion, which allows a sperm cell to swim. So obviously people with PCD, they have problems with fertility, as often their sperm cells and cilia in the fallopian tubes aren't working as they should be, and like George said, they have a lot of chronic infections in the chest. As illustrated as well, there is a cross section here which is called an axoneme. So basically it is the core of the cilia, and the axoneme contains many structures, but the ones we're going to be focusing on today is the outer dynenome, which is the little red structure here. And the outer dynenome and the inner dynenome work together to generate the sweeping motion of the cilia. But like I said, we're just going to focus on the outer dynenome today because the protein that DNA H5 encodes for is called the heavy chain 5, and that heavy chain 5 protein is present inside these little red outer dynenomes. So Katie will talk more about the genetics.
Okay, so the components of the cilia and flagella are encoded for by lots of different genes. But PCD-inducing mutations have been found in around 14 genes, and they make up around 60% of cases. PCD can also be caused by founder mutations, and what a founder mutation is, it's when more than one member of a separate population have the mutation, obviously it's passed on. So therefore it's seen quite often in members of the Amish community as well. But the mutation we're interested in today is found in DNA H5, which is found in the short arm of chromosome 5 at position 15.2. Now what a chromosome is a condensed bit of DNA which is wrapped around proteins called histones. And what this gene codes for is the heavy chain 5, which is found in the alpha-dyne arm that makes up the complex within cilia. And what happens is the building blocks of DNA, one of them changes. So when you get a base change from, say, a G to an A, this mutation, because 3 bases make up and code for 1 amino acid, a change in the base sequence from G to A results in a change of amino acid from the thymine to isoleucine. And what this does is it causes a termination signal. So when the protein is being formed, it stops it being formed prematurely, and therefore it leads to a non-functional protein like Nicole will demonstrate.
Yeah, so in terms of PCD, what actual protein is affected and what happens to it? So, like Katie said, the protein that is affected is the heavy chain 5, which is located in the alpha-dyne arm, and all the proteins within the cilia, so within the axoneme, all the proteins in that are made up of different subunits, which are encoded by a specific gene. So the subunits are like building blocks of proteins, and if one doesn't fit or one's not right or there's too many, etc., the protein won't function properly as it should do. So, as Katie said, because the gene is mutated, the DNA H5 gene is mutated, it means that the protein, the heavy chain 5 protein is truncated, which basically means it's shortened, so it stops being built sooner than it should do. So what happens when this, so what is the problem when this happens? Basically, once the protein is shortened, it causes the alpha-dyne arm to not do its function properly, because the shortened heavy chain 5 protein doesn't allow the alpha-dyne arm to generate force, which is what its function is. And the heavy chain 5 protein is a force-generating protein, so it uses the body's form of energy called ATP. It uses ATP to create force at the minus end of the protein. So this force then, in combination with and in cooperation with the inner dyne arm and all the other structures within the axoneme, it allows the cilia to bend back and forth and sweep, which is what it's supposed to do to enable things to move along it. So mucus, for example. And here we have an abnormal axoneme, which in this diagram here, the inner and outer dyne arms aren't present. This can happen in some types of PCD. In some types of PCD, the outer dyne arm is completely missing, which obviously causes a problem because then it's completely not able to bend. The cilia is completely not able to bend. With the DNA H5 mutation, the outer dyne arm is present, but it's a lot. The heavy chain 5 is a lot shorter than it should be. So the cilia do still move, but at not enough times as they should. So like per minute or per second or whatever. So so with the heavy chain 5, the force comes from the minus end of the protein, and it uses ATP to do that. So I think Nash is going to talk a bit more about this.
Thank you, Nicole. So obviously, if we now look at the cell and what the mutation does, so we know that cilia, which help to remove bacteria, infectious substance, and toxic substance from our inner respiratory tracts, they now become limited in their free movement, or they almost become immotile, which means you find a little bit of movement, but not enough to move mucus along. So this all comes down to the assembly of the dynein arms, which cannot form the full circle because they can't link on all the way around. So the cilia is made up of obviously central apparatus. You have the radial spokes which connect the surrounding to the centre, and then you have the dynein arms which link one another to each of the surrounding axonemes. So with the axoneme, what we see is it's sort of pushed forward using sort of like forward beating motion, and this is used with the fuel ATP. Now what it does is it breaks this molecule down into ADP and a phosphate group, which allows it to bind on to the neighbouring axoneme. Now to complete the cycle, it also sort of follows down towards the minus end, which causes the cilia to push over its neighbouring, the axoneme to push over in the towards the neighbouring axoneme, the beta tubule, and this gives the cilium the sort of beating motion back and forth. But because of the mutation, the loss of the autodyning arm is found, and therefore the full beating power and the full effective and powerful stroke isn't found there anymore. So the mutation which happens specifically to the autodyning arm by the mutation of DNAH5, and it truncates the protein in 28% of the cases, it doesn't manage to exactly assemble properly. So just a simple analogy to work off is like in hand. So in hand we have our different fingers, we have a thumb, which come together for a purpose to grasp something, to pick up something. Now the autodyning arm, if we try and imagine it as our thumb, although you would be able to pick up something using your fingers, it's not as efficient as using the thumb. So the autodyning arm plays an important role like the thumb, more or less. Another feature that's affected more specifically is the heavy chain. So if we look at the picture below the graph number 8, we can see that our alpha tubulin has 3 myosin heads. This has been identified in the Chlamydomonas green algae model. It's obviously been researched extensively. So they've identified these 3 heavy chains, but in humans it's only 2 have been identified, and with the DNAH5 mutation we get the loss or the truncated version of the gamma head. So if we look at the position of the gamma head, it's more or less positioned towards the lower, where it's sort of got the extended arm reaching more further out and causing more of a pull. Now because that gamma head's gone, we don't get that effective pull of the cilia, and the proper beating motion has gone missing. So this leaves us with the end result of the cilia being more or less stood still or not beating as properly, and the mucus stays sat within the inner linings and they just don't move from there. So George will hopefully link that back to the phenotype. Thank you, George.
Now, as described earlier, the 2 main symptoms of PCD that we're going to talk about today are infertility and the chronic respiratory tract infections. Now, with infertility, it does occur in both men and women. However, it is more common in men due to the flagella of the sperm cell not having the force to move the sperm cell at the required rate in order to meet the egg at the required time, as well as in women the egg not being able to be moved down the fallopian tube where it is accessible to the entering sperm cells. Because of this, the fusion of the 2 cannot occur, which will lead to the couple being infertile. As well as this, the chronic respiratory tract infections are caused due to the mucus that is being produced, which traps the bacteria and viruses we breathe in naturally. That cannot be removed from the body and stays within the windpipe. Therefore, it can lead to infections because of the people extremely coughing in order to remove the mucus, therefore leading to inflammation of the cells, as well as the bacteria in the actual mucus that's been collected invading the cells and causing it to to become infected. Now, as to summarise what we have talked about, PCD affects cilia and flagella. This mutation of DNA H5 causes a protein, dynein, to be shorter than is required, and because of this, these cilia do not have the correct force in order to move the way they should do, in order to perform the job they are needed to do. Therefore, these symptoms occur. And that is it. Thank you for watching. Have a good day.